So with your permission, sir. Good evening and welcome to the faculty and all the delegates who have logged in. I am Dr. Amit Borse, Head PMT and Medico Marketing Initiatives at IVA, a division of MCure Pharmaceuticals. MCure is #1 company in gynecologicals and a preferred partner in IVF. Materna is a core brand and is an epitome of quality and success. I, along with Mr. Nikhil Mehra, Divisional Sales Head, are very thankful to Dr. Rajan Vaidya, sir, for giving us the opportunity to host this event. And we are very thankful to Dr. Meenakshi Chaudhary ma'am to accept our invitation to join as a faculty. Birth of our first IVF baby marked the groundbreaking success in infertility management. On the eve of World IVF Day, a very important day in the field of ART, we are starting with a webinar series with yet another revolutionary advances in assisted reproduction treatments. Our theme would be from the first IVF baby to a 3-person baby. Before I go ahead, may I ask Mr. Nikhil Mehra to say a word or two?
Thank you very much, Dr. Amit. So very warm Sunday evening to everyone out over here. On behalf of AmCure, I am Nikhil Mehra, working as a division sales head. I would like to take this opportunity to thank Dr. Rajan S.
I have lost the screen.
Sorry, sir.
But I lost the picture. How do I go there?
So I think there is some technical issue at Dr. Vaidyan. What happened, sir?
I have lost the picture.
Mr. Hardik, can you please look into it?
Which picture, sir?
I would like to take this opportunity to thank Dr. Rajan S.
I have lost the screen.
Sorry, sir.
I have lost the picture. Can you hear me?
Yeah, you are audible, sir. You are audible.
But the screen is showing something else. It's join the live Zoom demo, get on the guest list, and something like that is coming.
You are already on the Zoom call with us, sir.
Yeah, but then why I'm blanked out sort of.
Can you see a Zoom icon at the bottom in your task?
Yeah.
Just click on that.
Zoom icon at the bottom.
Yeah, the blue color icon with the camera.
No.
In your taskbar, sir.
Yeah, I am.
Yeah, just click on that.
Yeah.
Now, can you see the Zoom window?
No. I am not seeing the Zoom window.
Okay. Can you just click Alter and Tab?
Alter and
Tab together. So you will see a couple of options on the screen.
Alter and what you said?
Tab, T-A-B.
T-A-B.
Yeah.
I have to type T-A-B.
No,
sir.
On your keyboard, there is a tab button. On the left hand side, third button from the top.
Should I re-log in?
No, sir. Sir, we can see you. You are here.
I can't see
Dr. Amit, can I just stop sharing your screen? Then I think it will be easy for sir.
Yeah, please.
Now it's there. Yeah, perfect.
So it's fine. So this is only about, I'm sharing the screen. So we'll get you live. Not a problem, sir.
Okay.
So I will.
Right.
So let us, Nikhil ji, please.
Yeah. So, so on behalf of AmCure, we would like to thank Dr. Rajan S. sir for giving us a wonderful opportunity for, hosting international icons of IVF. And we are thrilled to have Dr. Meenakshi Chaudhary from UK, our guest speaker, for the very first event, which is International Icon. And today our international icon is Dr. Meenakshi Chaudhary. So thank you so much, ma'am, for having, being over here. And we all know that it's a wonderful day today. So it's a World IVF Day and, we are kickstarting this international event. And we will be looking forward for more such events, sir. And a wonderful session today. So thank you so much. I'll just quickly take a minute to introduce about AmCure. So AmCure is a company which is available in more than 70 countries worldwide. We are the #1 gynecology company in the Indian market and the most preferred partner in the field of fertility management. So thank you so much, ma'am. Thank you so much, Rajan as Vaidya sir. And over to you, Dr. Amit. Thank you.
Thank you so much for that introduction, Nikhilji. So may I take this opportunity to introduce our beloved Dr. Rajan Vaidya sir. Sir is an honorary consultant at Department of Assisted Reproduction, Naurozji Wadia IVF Center, Parel, Mumbai. He's an international and national faculty at ART conferences. He has his own center with the name of Ankur Fertility and Endoscopy Center, Kalyan, and Venkatesh Hospital and Nursing Home, Kalyan. He has been with peer review journals of he has been at a peer review journal for human reproduction and embryology. He is member and affiliate to many organizations. We welcome you, sir, and may I now request you to speak a few words and kickstart with the series.
Good evening, everybody. Thank you, Dr. Amit, for that kind introduction. And thank you, everybody. Good evening, everybody here in India. Good evening, everybody here in India. And good morning, everybody, all the viewers in the United Kingdom and Europe. And hello to everyone from other parts of the world. As we all know very well, that it was on this day 43 years ago that the world's first IVF baby was born. It was indeed a groundbreaking success in the management of infertility and science of human reproduction. Right since then, however, there have been unrelenting quest for bettering the reproductive outcomes and circumventing certain inheritable diseases using newer and newer technologies. Mitochondrial replacement therapy for mitochondrial genetic diseases constitutes one such spearheading development in the prevention of inheritance of mitochondrial diseases. Recent successes in this technology has been encouraging and holds a great promise to be used more frequently and in many more countries. Such cutting-edge technologies can improve the lives of the patients and families who have been afflicted with such mitochondrial diseases which have devastating consequences. To know more about mitochondrial replacement therapy, I request Dr. Meenakshi Chaudhary, who is our speaker for today and a very renowned international faculty who has done a pioneering work in the field of mitochondrial replacement therapy. I request Dr. Meenakshi Chaudhary to please start a talk and enlighten us on this wonderful novel treatment for inherited mitochondrial diseases. Dr. Meenakshi, over to you.
Thank you so much, Dr. Rajan Vaidya, and thank you, MCure, for giving me this opportunity to present today. I'm just hoping that I will be able to share my screen in the right way, rather than the glitches sometimes which I encounter. Okay. Right. Is it working absolutely fine, the screen?
Yes.
Okay. Right. Good evening, everyone in India, and hello to everyone else. Thank you so much for this brilliant opportunity to present. My talk is on the first IVF baby to a 3% baby, the move, the technological advances in that. And I'm very thankful to, as I said already, to Dr. Rajan Vaidya for approaching me for this, as well as MCure for hosting this series. I don't have any conflict of interest, except that I do express my opinions on social media, especially Twitter at Meena Fertility. Also, one disclaimer I would like to make is that this may muddle your brains up, and because it's full of scientific jargon, there is a possibility whenever I give talks, I've given these talks in Japan, Greece, Europe, America, lots of countries, including UK, I shouldn't forget UK and India, is that whenever I give, a lot of people's jaw will be dropping, and sometimes it will be in awe of the science, and other times because they've fallen asleep and they're drooling, and that's why their jaws are dropping. So I don't know which group you will belong to, but let's start our talk. So the quest to overcome inheritance of genetic diseases first begins with the understanding a bit about the main players. So my talk will be specifically focusing on the mitochondrial diseases and its inheritance pattern. We will discuss about the reproductive options available, as well as we'll discuss the advanced ART techniques to minimize the risk that we had to encounter and develop and process and design and plan in relation to science, ethics and regulation, and the 3% baby. And we will also touch upon, lastly, the future. So, Newcastle is in the UK. It was the first in the world to legalize, UK was the first country in the world to legalize mitochondrial donation therapies. And that's where Newcastle is. It's in the northeast of England, and you can see just over there. So what do couples want? We know that all couples want is a miracle, a fairy godmother to sprinkle some baby dust, and especially for these mitochondrial DNA disorders women, and give them a healthy baby free of the curse of inheriting the deadly disease which they have got. So this is the crux of my talk today, and all they want is a healthy baby, and let's see if we can help them have that. It's not about engineered and non-engineered humans, ethical debate, but more about the science behind it. This is my first baby. So she wasn't a miracle. She just came without needing any extra support, but she is now a miracle after her birth, I would say. So we all know, we all are in the scientific field, and we know about the pre-implantation development of the embryos. From a zygote initial stage to the blastocyst, where it is of 100 to 200 cells. So my talk will be focusing more about this pre-implantation stage in a quest to the 3% baby. We all are 2G humans. And what I mean by 2G humans is that we do have two genomes, the nuclear genome that comes from the egg and the sperm. So when the egg and the sperm unite together, you can see the two pronuclei which is formed once the egg is fertilized, one from the male pronucleus and one from the female. And this constitutes our nuclear DNA. So this is our one genome, that is a nuclear DNA. However, we do have another genome, and that is the mitochondrial DNA, which is present in the cytoplasm of the egg. And all of our mitochondrial DNA is solely inherited from the maternal line. The sperm also has got mitochondrial DNA, but when the sperm enters the egg and fertilizes the egg, that mitochondrial DNA is no longer available to constitute in a zygote. So we have got two genomes, one from the nuclear DNA and one from the mitochondrial DNA. Or in other words, one is the maternal genome and one is the paternal genome. So coming on to knowing a bit more about mitochondria, because you can't start to talk about mitochondrial donation and transfer without first focusing on what mitochondria is and what it is role. It is called the powerhouse of a cell, or the batteries. So what we know is that more than 90% of our cellular energy is produced by the mitochondria. So it is really the powering boost that it provides to all our cells and organs to function. Now, mitochondrial genome is a very tiny genome. We know that we read about the 100,000 genome project and all that our human genome sequencing has been done. But mitochondrial genome is an extremely small genome, and it consists of only 37 genes. There are multiple copies of mitochondrial DNA in one oocyte, and it can vary between 200,000 to 300,000 copies. What we know is that human genome itself contains of 3.3 billion base pairs, but mitochondrial genome is quite small, maybe around 16,000 base pairs. So you can imagine there's a huge difference between mitochondrial and nuclear genome. And not like the nuclear genome, where once you have the nuclear DNA, it is the same in all cells of your body. Sometimes in mosaic, you may have different proportion of quantities, but otherwise it's the same. But when we talk about mitochondrial DNA or the mitochondrial genome, there is a heteroplasmy. So what do we understand by heteroplasmy? It tells us about the wide variation in the mitochondrial DNA mutation loads in different cells. So the urine sample, the heart cells, muscle cells, the lungs, or in the eggs, they will have different load between each cell. So that's why it can be either very low mutation, so the person may have no heart problems, or it may be very high mutation load in the kidney, so they may have a lot of kidney problems, or it may be homoplasmic, where all the cells are severely affected. So if you look at the graph, which I've displayed, you can see the first one. The green are the healthy mitochondria. The red ones are the mutated or abnormal mitochondria. So the first one is a completely normal with no heteroplasmy or homoplasmy seen. So they're all healthy mitochondria, what we call as a wild type mitochondria. The second one has got a mixture, and the cells are having mixture of healthy and mitochondrial. And depending on what part of the organs get what load, it will differ. The last one is where all the cells are affected and it's homoplasmic. In that case, any child born to these mothers will be affected. Oocytes usually may have a mutation load that may vary between 0 to 45%. So we talk about, as I mentioned, about different variation of the mitochondrial load in different organ systems. And you can see mitochondria is essential for all the different parts, so all the 3 germ layers and the different parts formed by that. And that can contribute to the health problems that a person would encounter. And what diseases does mitochondrial DNA mutations cause? So it can cause mostly the neurological-based diseases, as highlighted on the right side of the screen, and non-neurological based, like respiratory failure, cardiomyopathy, liver failure, and diabetes and thyroid disorders. So there are several implications of that, and it can be life-threatening. Most important, what one needs to understand is that the prevalence, it is not a very common condition. The prevalence is around 1 in 5,000. It is not common, but it is seen. At Newcastle, we have got a specialized sort of a national, supra-regional referral service for mitochondrial DNA disease patients. So when someone has got reproductive mitochondrial DNA disease, it can cause heteroplasmy. So I have deliberately for this talk blocked the family, although the family had given us permission to display their pictures, but you can see the silhouette of them. So the mom had 38% load of the mitochondrial DNA mutations. However, she herself did not have any known clinical symptoms. The diagnosis was made when her son, who was 7 years of age, he died with multi-organ failure as listed on the right-hand side. The son had 78% load of mitochondrial DNA mutation. That's when the mom got tested and came to know that she had 38% mutation load. Subsequent to that, she has had, before her son died, she had a daughter born and she's got 0% load. So you can see the huge heteroplasmy, and that tells you that some babies will be absolutely fine and others may be affected, and the family may have to go through the turmoil of losing a child because it can be life-threatening and life-debilitating. And currently, there is no cure to result in a healthy child and future generations for this. A maternal line inheritance means that it will be passed on from mother to both sons and daughters, but the daughters will pass it on to future generations. So can we do anything to help minimize the risk of inheritance of these mitochondrial disorders using the assisted reproductive technologies? So this was what we were trying to look into and see if we could fix any problems that are with the reproductive options we have. So these are the current reproductive options which is available in most of the countries. One is the preconception option, which is the PGD. Not every center would have the PGD option, but PGD for mitochondrial DNA disorders, so that you can test for those, not the nuclear diseases or the point mutations we are talking about. So that's why I've not put it as PGT, because the for mitochondrial DNA disorders is still referred to as pre-implantation genetic diagnosis. Post-conception, if a mom doesn't want to go through fertility treatment and because they do not have any known fertility problems conceiving themselves, they could conceive and then opt for prenatal diagnosis, either by chorionic villus sampling or post-conception by amniocentesis, and assessing whether they have got the mitochondrial DNA load mutation copies and looking at that. And if it is a high load, then the ethical dilemma of whether they are willing to terminate the pregnancy or not. The third alternative option that was available to everyone was the egg donation. That is, just overcome the barrier of these mitochondrial DNA mutation errors in the cytoplasm and replace the whole egg. The con of this is that, unfortunately, the mom would lose having her genetically linked child as well. So a little bit more about PGD. So we get some idea as to what PGD for mitochondrial DNA disease encounters. In the UK, the regulatory body is known as the Human Fertilization Embryology Authority, and the HFEA has to give license to any center to do PGD or PGT. For any diseases, so if we have got a license for doing PGD for mitochondrial DNA disease, but if someone has got, say, Huntington's and we wanted to do PGD for that, we do not have license for that, so we won't be able to offer that. So there are centers where they have to be licensed for every condition. Some may be licensed for nuclear DNA diseases. So the evidence to date indicates that PGD provides a promising risk reduction strategy for affected families, so it can reduce the risk. However, one needs to understand that when we do PGD, where we take few cells out, analyze them, and then replace that embryo if everything is fine, it is not appropriate for everyone, especially if the women are homoplasmic, as I was talking earlier about what homoplasmic means. So if all the DNA in the cells, mitochondrial DNA, are affected and mutated, then it will not be an option for those women because they will definitely produce high mutation load embryos. And the other is it's not an option for women who are heteroplasmic, but consistently they produce embryos with high mutation loads. So we are doing PGD, but we do not have a healthy embryo to put back in for that woman to reduce the risk of transmission. So in those cases, PGD is not an answer for risk reduction. So are there alternative strategies? Can we uncouple the inheritance of the nuclear DNA from the mitochondrial DNA to reduce this high mutation load embryos and give the mom a chance of having a healthy baby? So this was what we set out to achieve. And the answer we got was through new technological advances by mitochondrial replacement therapies. So as I mentioned earlier, currently there is no cure to result in a healthy child and future generations for these moms. However, this promising new technique has given us hope to detect it early, fix it early, before conception, so that children born to these women from affected families are healthy, disease-free, and we can break the chain of inheritance, and we're not leaving it on the gamble of healthy embryo or not healthy embryo of natural conceptions or with PGD. So we are trying to take it away from the Russian roulette. So as one of the editor-in-chief of Escherichia Journal Human Reproduction put it in his editorial about the therapeutic illusion. So we are talking about, so we have thought about this concept. Let's see if we can uncouple and do this mitochondrial replacement therapy, but will it work or will it be like a therapeutic illusion? Now, what we understand by this therapeutic illusion concept is that we ascribe a successful treatment to our intervention. So a lot of people talk about rejuvenation therapies or different techniques, which doesn't have strong evidence base to support it. But if we have one successful treatment, we allocate that success to that treatment procedure or the intervention we used. However, it could be that is actually most of them would have gone on to have a pregnancy or success regardless of the intervention or the treatment. And this is classed as therapeutic illusion or the Santa Claus in our fertility clinic. So that's how we have to consider whether it's really going to work or if we have one live birth, this just would have happened anyway, regardless of that. So now coming on to a bit more of science. So what are the different ways of mitochondrial replacement therapies that one can do? So one of the ways is the pronuclear transfer. Now, pronuclear transfer is, so as I mentioned earlier in my talk, that when the male pronucleus or the male genome, the 23 chromosome pair, pairs with the female, the maternal genome, and the egg fertilizes, it forms the pronucleus. So at that stage, you transfer that. You can see there is a donor oocyte, which is at pronucleus stage, and there is a patient oocyte at pronucleus stage. So we take this nuclear DNA out from both of them. So they're being extrapolated out of that. Once they're taken out, we take the healthy cytoplasm of the donor oocyte and insert the nuclear DNA, the pronucleus we have taken out from the patient oocyte into that. So now they have got the healthy mitochondria within that with the nuclear genome from the parents who want to have the baby genetically linked to them. So this is the reconstituted zygote that forms. So there have been publications in mouse as well in relation to success of or the feasibility of this study, as well as in human, and this is from our center in the Nature. The other technique is the metaphase 2 spindle transfer. In that, we do not fertilize the eggs first. We just use the oocyte at metaphase 2. So once you have collected those oocytes, you identify the spindle, you take the spindle out from both the donor oocyte and the patient oocyte, and then replace the patient spindle into the egg, and then go on to fertilizing it with the sperm and resulting in a reconstituted zygote. But there is an issue in this with mitochondrial spindle transfer. And the issue with that is that you can't see a spindle with naked eye. So for looking at the spindle, you require polarized light by refrigerants to visualize the spindle. And so you have to use the dark grounds. So it's not possible to detect or visualize these chromosomes. And because the spindle is so delicate, and it's a very dynamic state when the meiosis resumption is taking place, that it is sensitive to disruption and can result in chromosome segregation errors if you're doing that. So one has to be careful. So there have been publications in relation to this technique as well. And the last technique is about the polar body transfer. So in that, what one can do is, once the mature oocyte is collected, you replace the polar body. So you take the polar body out, take the polar body and the MST from the donor oocyte and replace that polar body into the donor oocyte, and then reconstitute, activate that oocyte, and once it's formed, do the ICSI with the sperm and create a reconstituted zygote. There is the concept with polar body is that we may be able to get two, if the patient has low oocytes, so then one, we can use the spindle transfer, and the polar body extruded can be used for creating a separate oocyte as well with the donor oocyte. So we may get two embryos from one. So it looks quite promising. However, the success of this technique is as yet not done in humans. And in the end, all these three techniques do result in reconstituted zygote, which may be capable of resulting in a baby. So now this is a video from our pronuclear transfer technique. So you can see the needle going in and the two pronucleus were seen. One pronucleus is taken out, and you would see that, so that's the one pronucleus. Now the remaining pronucleus, you can see the needle is going to go back in to get it out. And initially we would take both pronucleus out together, but that causes problem as well, because you may get a lot more of patient cytoplasm, which we don't want. And then that could result in a problem as well. So both the pronucleus are taken out. Now this is, when the pronucleus is taken out, they're called as karyoplast. And then they're briefly exposed to sort of a viral media with the HPJ so as to help it bind into the other egg. So it's a viral envelope protein. And you can see now that we are inserting into an enucleated oocyte, the donor oocyte, both that. And you will see sometimes these oocytes need a little nudge, fertilized oocytes, and that nudge will help them to combine the nucleus, both the nucleus into the cytoplasm, and help with the division subsequently. So the key issues to be addressed for us to reach that stage, the video which you just saw, is to address the science. That was one of the key issues we wanted to address. And from the science perspective, we need to look at, will it work? Is it efficient? And is it safe? Because these are the three things we need to know. And we can't go on directly employing the technique without, in clinical translation, without answering those questions. And the two stalwarts of this is Louis Heaslop, who is our senior embryologist and manager, who did all these scientific experiments in the lab to develop the techniques, to troubleshoot and all, under the guidance and supervision of Professor Mary Herbert, who is embryology director and professor in reproductive biology at Newcastle Fertility Center. So these are the two scientists who did the work in the laboratory for that. So it is a teamwork, but this is the work done by them, and it wouldn't have been possible without that. So for doing this research, we need some oocytes, and we need some normally fertilized oocytes. So there is a possibility that one slide is missing, or it may come up later on. So we did try doing with different types of oocytes. The normally fertilized oocytes we sourced from was by egg sharing, where some women donate some of their eggs for research purposes. I published that in the Cell Stem Cell Journal, that data, and non-patient egg donors, where healthy volunteers, women come forward to donate their eggs for this valuable research. And nowadays, almost 80% of our oocytes come from this source. And there is a compensation offered to these healthy volunteers who come forward, which has been approved by the HFEA. So to getting started, we had to encounter three main challenges. So I'm going to talk about it because we're talking about the journey towards the 3% baby from the first IVF baby who was born years ago now. And that time also there were a lot of challenges. You know that MRC refused to fund that when they set out to do the first IVF. And subsequently, they went on to do it and got the first baby. And there was a lot of ethical hype that time as well. And we also encountered our fair share. So let's talk about the research material, and then we'll go on to regulatory and legal and technical aspects of challenges. So the first challenge for research material was that, as I said, we need to do this research, we need normally fertilized eggs available for research, but who will provide those eggs? It's not common for us to get those eggs. So we set out by first doing this proof of principle experiments by utilizing abnormally fertilized eggs. So by abnormally fertilized eggs are those when you check the day after egg collection and insemination, the oocytes, you would see that some of them may be 3 pronuclear or 1 pronuclear. So those ones which will be otherwise be discarded and not taken into consideration for transfer were the fertilized oocytes which we had available. But more importantly, apart from the research material challenge was the regulatory and legal. So the HFEA, as I mentioned, governs the UK law, and all centers have to be licensed by them. So as per the HFEA Act of 1990, under this license, one cannot authorize the altering the genetic makeup of any cell while it forms part of an embryo. And fertilized oocyte is classed as an embryo. So would the genetic structure be altered by us doing this procedure of pronuclear transfer? So that's what we needed to address. And so we had a lot of deliberations with the regulators and scientists and everyone. And the conclusion after a lot of deliberation was that pronuclear transfer would generate new combinations of nuclear and mitochondrial DNA, but the actual genetic structure, the nuclear DNA, which gives us all our characteristics, our identity, will not be altered. So one thing one needs to understand clearly is that, as I mentioned, a mitochondrial DNA is like the powerhouse. So the best way to describe it is like the battery. So if you've got a camera and the batteries are not working properly, you replace the battery and the camera starts functioning again. So that's what the mitochondria does. So it provides the energy to those cells, to other body organs and body tissue to function. But how the camera would function, the quality of the resolution of the pictures and images will all depend on the camera features, not on the battery. So if the camera is a black and white camera, an old type camera, it will not give you the clear resolution. But if you've got a very high megapixel, those SLR type cameras, then you will get brilliant pictures. So that comes from the nuclear DNA in terms of the characteristics and how the other identity would come from that. So that's the difference one needs to know. And rightfully, the ethical body, the regulatory body and all agreed that we were not altering the nuclear DNA because no one had ever asked that question, does mitochondrial DNA constitute to this genetic structure, even though it contains less than 0.3% of our whole genetic makeup? And they rightfully came that it will not be altered. And they gave us permission or license to carry out research. Even research in UK has to have a license, and I lead on that research. So I'm the legally responsible. So if anything goes wrong in a research laboratory, I will be the person who will go to the jail and prison and serve under criminal offense. So the third challenge we had to encounter was the technical challenge. So one was the enucleation. So can we take it out without disrupting the success of that egg to the fertilized embryo to develop? So we had to use chemical inhibitors. It's not easy to pull out a pronucleus. So you have done PGD, PGT, or ICSI, and you put a small bore needle in, take a cell out. But with this, the pronucleus are really tightly stuck together, very stuck with and you have to use some chemical inhibitors to depolarize the actin and microtubule, the skeletal network, otherwise you can't pull it out. And the laser-induced hole in zona pellucida you make, it can make the cytoplasm leak out as well because it is not, the needle is not the same size as we use for ICSI, it's a wider bore needle. So that will be a big gap that would be left. And lastly, the pronucleus is surrounded by some cytoplasm. So however careful you are, as I said, we initially started with removing 2 pronucleus together rather than going in and out of the oocyte. But we realized that it brought in a lot more cytoplasm, and along with that, it will bring the abnormal mutated mitochondria as well. So we had to be very careful and do it and minimize that. And in the video, you might have seen us doing that pinching technique to try and get rid of the excess cytoplasm. The other step, technical challenge that we faced was the fusion. So when you are putting that karyoplast that you've taken out into an enucleated zygote, will that fuse together? And electrical fusion, which is commonly used in mouse experiments, was not working for this technique when we tried, and it was a big failure. So we had to think about alternative way, and that's when we considered using the inactivated centavirus, the HVJE viral envelope protein. So they were coated in that, and that helps it to fuse. So that's the thing we use, but one needs to understand that sometimes these things will not be CE marked or licensed for this. So when it comes to clinical translation, that would be another challenge to get it approved or to inform the patients. So that was another challenge we had to overcome before getting it translated into clinical practice. So, so from our abnormally fertilized oocytes, as I said, that was our first source. We looked at the proof of concept, whether is it possible to do it. So we did find that it did result in zygotes, which developed, divided and all that. So it was technically feasible. We could take it out, replace it into a different oocyte, and it was compatible with the development to blastocyst stage. But if you look at the graph, you can see the blastocyst rate formation was extremely low compared to the unmanipulated controls. So controls had a higher blastocyst rate compared to this one. So this clinically, it may not be suitable if you're going to lose that many oocytes when we come to clinical translation. We didn't know whether that low blastocyst development rate was because of using abnormally fertilized eggs, and whether that has a different dynamics within it and the staging that prevented them developing. The other thing which we confirmed with the proof of concept experiment, that the levels of mitochondrial DNA carryover from the patient oocyte was significantly low. So hooray, that was a big success. And as I mentioned earlier, this was published in, which is a very high-ranking journal for those who are not into academic field. It was published in 2010. So you can see it's been many years of work to get to that stage from a first license in 2005, 2006 to do the research, 5 years to get to the stage of publishing the proof of concept experiment. However, as I mentioned earlier, meaningful tests of safety and efficacy require supply of normally fertilized eggs. And as I displayed earlier, the slide, which was misplaced, is that we initially started off with egg share. So initially we started off not with egg sharing, actually. Women who were going through IVF treatment, they were asked if they would be willing to, if they produce 12 or more eggs, will they be willing to spare 2 of their eggs? Then egg number 13 and 14 were donated for research. So we started off with those normally fertilized eggs. Subsequently, we realized that was not enough. There was scarcity of the dose, the number of oocytes we were getting for research, and there were hardly any progress made in the scientific experiments. So that was also ethically approved, because any research has to be ethically approved in UK. Then we went on to egg sharing, where we, as a benefit in kind, women who volunteered to share half of their eggs for research were compensated by reduction in the cost of the treatment cycle. So this was an option for those who were self-funding their IVF cycle in UK, in Newcastle at least. Couple can have 3 cycles on the NHS. So those couples will not go ahead using that technique. So one has to balance everything out. Then again, that did help progress the science further, but again, there was a challenge. And then finally, we went on to egg donation for research, where we were given permission to approach healthy volunteers if they wanted to donate eggs for research. And that was helped by these 2 landmark reports, well, one in 2000, both around 2011. One was from the HFEA, the new policy on compensation. Prior to 2012, actually, the reports were published in 2011. And then 2012, the HFEA approved that one can compensate. So before that, one could not compensate any women donating their eggs, even for treatment purposes. So no compensation at all. But following this report, they looked at donating for treatment. There was another report, as well as for research as well, safeguarding donors. Can we donate for research, and can we compensate that? And Nuffield Council on Bioethics is a national body which looks at all these ethical dilemmas and questions relating to organ donation, human bodies, and everything else in relation to advances and technological advances, and genome editing as well, their latest one they were reporting on. So they concluded that, yes, one can consider donating, compensating donors for research, and there was a set limit. So for treatment purposes, upper limit is £750 in the UK, which will come to around, maybe maximum around 75,000 rupees per donor, per donation cycle. And so that made a difference, that we could then approach, after seeking ethical approval, women to donate eggs for research. It caused an influx of women expressing interest, but from those women who express interest, you need to know that financial compensation is not everything. Only 5% of those who made inquiries went on to do successful egg donation, or went on to proceed with egg donation. So there was a lot of dropout at earlier stage. And one has to look at potential barriers and facilitators why women do not consider to proceed. That's a different talk altogether, which I do give as well, but not for this. Otherwise, we'll run out of time. So we started our PNT using normally fertilized zygotes, because we had this ready supply now of fresh oocytes that we could time it properly, because the abnormally fertilized oocytes, we were getting it much later in the time period and all. And we started doing it. And we realized that, so we would get these oocytes, fertilize them, and then 16 to 20 hours after post-insemination, we will replace that is do the PNT technique and look at the development. But the huge problem with this technique was 40% of the reconstituted zygotes did not even survive. And that's a huge loss of this precious resource. If you're doing a treatment for a patient, you just cannot afford to lose 40% to even start developing. So we had to address that issue, and we had reached an end of the tunnel, like we were in the dark space. We didn't know what to do. How can we resolve this issue? And that was a moment of despair for all of us in the scientific field, because we have been working on this research for such a long time. And here we go, we were back to square one on the drawing board. But at end of every dark tunnel, there is light. And this is our Newcastle on a bright nighttime when the sky is clear and you can see it's beautiful or lit up. And that's what happens sometimes in science as well. You start feeling in the dark and suddenly a light bulb shines and you can see your way through and you can proceed further. So this was our light and we started troubleshooting. So what can we do to change that? And what we thought is that probably because we were doing the PNT process around 16 to 20 hours post-fertilization, maybe there wasn't sufficient time to recover between PNT and the first mitotic division that happens, which results in two-cell embryo. And the dynamics, there are lots going on into that which we still don't know. And maybe the dynamics to reconstitute, reconfigure, and develop is not possible. So what if we switch our PNT process earlier? So what about us doing it at 8 hours post-fertilization, and will it work? So that's what we thought of doing, ePNT or early PNT transfer. So as soon as they're formed, get it out. So that's what we did next. And hooray, we did encounter success. Our survival rate of normally fertilized zygotes or the reconstituted zygotes increased significantly. So with the late PNT, you can see 40% were not surviving, but we almost got to over 90% with this early PNT technique. So we were quite pleased with this and we thought that's the way forward and that's what we will be doing going forward. However, Louis, our scientist, had a tough time because you can imagine 8 to 10 hours when you're doing it, you have to come in the middle of the night. So some nights you will be here in the laboratory at 2 o'clock, 3 o'clock at night time, doing the PNT process, trying to get this. So a lot of endless hours have gone in without extra pay, definitely. So we also had to troubleshoot and maximize the information we get, because if you're going to go to the HFEA and to the scientific expert panel to say, can we do it in treatment for patients now? They would say, have you looked at this? Because you're taking the nucleus out, has it caused increased risk of aneuploidy? Have you checked that? What about the mitochondrial DNA carryover? What was it in your reconstituted zygotes? And what about the gene expression? Has the gene expression been altered? The epigenetics of it and everything. We had to look at all those parameters and not forgetting the blastocyst development rate. So if you look at the blastocyst development rate, the gray one is the control group, that is, no manipulation has taken place. The blue bar is the autologous, which means that 2 eggs from the same donor oocyte, same donor, has been reswitched. So we have got 2 PN embryos from the same person, and we have just cross-pollinized them, you can say, as they switched over the PNT. And the third one is heterologous, and you can see that if we do it between 2 different persons, the success rate of blastocyst development was lower. So we need to know that there are some more dynamics that is happening, which we don't have answers to. And it's not same as the autologous transfer. And there might be haplotyping playing a role into it and a lot of other things. So you can see, but it was still acceptable with around 30-40% blastocyst development rate. So it was showing promise. So from our experiment, what we noticed, the summary of our ePNT blastocysts from this healthy normally fertilized oocytes was that there was no detectable effect on the incidence of aneuploidy. There was no concerns noted with the expression of genes encoded either by the nuclear or the mitochondrial genome. So we concluded that it is likely that this early PNT technique is compatible with the establishment of viable pregnancies, because that's all one can go in research lab. You can't create viable pregnancies with that. But the key question then is, but can it prevent the transmission of mitochondrial DNA disease? Because that's what we have set out to do. And to know a bit about it, so mutation load, as we discussed, varies, and the risk of disease and inheritance pattern changes, alters with the mitochondrial DNA carryover. So if the mitochondrial DNA mutation load is, say, less than 18%, then there is a low probability of disease symptoms. So the person themselves will have either no symptoms or very mild symptoms. So that will be reassuring. That will be okay. That means we are giving them a healthy life. But to prevent inheritance of transmission to the next generation, the load has to be less than 5%. And that's what we were set out aiming to achieve. So this is our second Nature publication in 2016, and the highlighted bit, you can see that we noticed that with our technique, when we did that, we found that less than 2% of the mitochondrial DNA carryover was noted. So that was a huge success because we did manage to reach the stage of less than 5%. However, one needs to know that some of the blastocysts, when they were cultured, they got up to a little bit higher in some cells, up to 5%, but still quite low. But that's something one needs to keep an eye on. So you just can't say, yes, it can prevent it, but that is at present, based on that conversion in the laboratory, we cannot guarantee with the subsequent divisions of the embryos that it will. So it will minimize the risk of inheritance, but whether it will definitely prevent fully, that only time will tell. But whatever we could do from research point of view to take it into clinical practice, we had done. So from this experiment, we concluded that, yes, it is technically feasible. It is, even with the normally fertilized oocytes, we can all achieve that. And this was published in 2016. So from that proof of concept experiments from abnormally fertilized to normally fertilized, we achieved identifying all this. So we set out to address three things. Will it work? Yes, it was working. Is it efficient? Yes, we were getting blastocyst development rate. And is it safe? Yes, the mitochondrial DNA carryover was much lower. So we did manage to do all this. But as I mentioned, we cannot say till we do look at the babies born whether it will prevent further transmission in future generations. So it was in turn, you can see 2010 was our first publication, 2016 was our next Nature publication. So it was more than a decade of hard work in this mitochondrial donation for research field, which culminated into 2 Nature publications and in highly reputed journal, giving us the international recognition as well. But along with that, one needs to know that regulatory and ethical aspects one should not forget. Is it legal? Is it ethically acceptable? As I said, we did do everything legally because we had to seek permission. But from ethical point of view, when we first set out doing it and trying to change the law, we were called as the Frankenstein's monsters because we were trying to create hybrid from 2 women, and that's how the press labeled us. So we had to, as we know, with scientific vigor comes media frenzy and ethical hype, and we had to marry the ethics with the technology. And that's the gist of all this presentation and all that we tried to convincingly set the scene going forward for everyone. So was it a slippery slope with ethical debate? And this was heavily debated in Parliament, in open public consultations by Nuffield Council on Bioethics. And what they concluded was that the committee looked at the mitochondrial donor status, because no one has been a mitochondrial donor before. So what would be her role as will she be linked as a genetic donor or like what we have in UK? So at present, altruistic donors who donate their eggs for treatment, they can be identified when the child is 18 years of age. But what about mitochondrial donor? Would they have the same status as that? That means they will be identifiable, but their nuclear DNA is not used. Or will they be more like tissue or organ donor where they will remain anonymous? So the law has stated that they will be anonymous, but that was the debate they need to look at. Any resultant child, will they have an identity conflict or self-identity conflict? Is it a germline therapy and is it okay to do this? And the safety and efficacy, as I mentioned, was uncertain. And is there any risk to resultant child? So all this was debated, but sensibly they concluded that it may result in significant health benefits to the progenies of the sufferers of mitochondrial DNA disease. And if they're proven to be acceptably safe and effective on balance, it would be ethical for families wishing to use them to do so. And this should, however, be subject to offer of an appropriate level of information and support, and not forgetting long-term follow-up as well. So from the legal perspective, the HFEA Parliament perspective, science had to progress in parallel with the legal and regulatory system. As you saw, that it took us more than 10 years to do the science. And if we would have started our legal journey to get it translated for clinical practice after our science was proven, we would be another 10 years back later. So we started both the process simultaneously as shown here. So you can see we started the journey very early on. And then along with that, on the right-hand side, you can see that we are publishing papers, and the HFA, the law is getting approved, looked at, and they are passing the regulations, and they are convening expert panel, the HFA, looking into it over the period of time. They reconvene to discuss after our findings are published. And then finally, in 2015, the regulations came into force that, yes, we can use this. This should be allowed for treatment purposes. But that is just not allowed. That's just the regulation. The laws were written up. However, after that, the HFA had to reconvene, look at the cautious rules, and then they recommended the cautious use of this technique for high-risk cases only. They do not approve all cases, only if the women are homoplasmic. Every individual case needs to be sent to them, or if the PGD continuously results in high embryo mutation loads, then they would consider, otherwise they can decline the request as well for individual cases. And subsequent to that, the first treatment license was not offered till March 2017. So even though the regulations was passed by the Parliament in 2005, you can see it took longer than that for us to proceed with it. So far, around 15-20 license applications have been submitted to the HFE and being approved to offer mitochondrial donation treatment for women with high risk for serious mitochondrial DNA disease mutation. However, we did have few which have been turned down as well. So one needs to know that. And one needs to know that it is only permitted in cases where there is a risk for serious mitochondrial DNA disease. And research so far does give a promising outlook. And it's just the beginning. There is need for ongoing research and a need for long-term follow-up needed. So it cannot be used for any other purpose apart from the mitochondrial disease mutation. So that's the key part of it. I'm sorry, I can't disclose any outcome of the treatment process as such because of the confidentiality. Till the patients give us permission and the HFE gives us permission, we won't be able to share it right now, but we are hopeful that the permission will come soon in near future for us to publish our findings of the clinical practice and the data from that. So we shouldn't forget the real heroes, the people in all this planning, designing, and the work that has gone behind. We should not forget the real heroes, and these are the girl power. How did we reach to the clinical treatment, the story of bench to bedside, and the women who worked behind that, and those are the ones, the mitochondrial donors, the women who came forward to donate their eggs for research purposes and powered the study. So powering the clinical translation, the mitochondrial donors, women who are donating the eggs for research and for clinical treatment are not only giving a gift of health to just one child, but they're also providing that to future generations of children to come, as you can see. So we are hoping that it will get passed on, and we would have lots and lots of healthy babies who will become healthy adults and healthy females who will not then pass on to future generations. But coming on to the $1 million question, which everyone asks us from all over the centers, all over the world, who wants to come to Newcastle to look into this, is can we use this technology to prevent the age-related decline in the oocyte quality and rescue those women by replacing their mitochondria and improve IVF success rate? So what I need to tell for that is that we have done a lot of research for more than 10 years to prove this for mitochondrial DNA disease mutation. And in that, we have also done experiments in mouse, as well as we have got a multi-million funded project, the GermAge, where we are looking at oocytes and aging process. And in that, the mutation load and heteroplasmy in chromosomes in women, older women, is no different to the heteroplasmy, the mutated mitochondria, to younger women. And also, this is shown that women who have got heteroplasmy or mitochondrial DNA disease themselves, they do not have a fertility problem, because if that was the case, they will not be able to conceive naturally. And they do conceive naturally. They have babies naturally without any problems, fertility problems. So that's why the role of mutated mitochondria resulting in answering those questions for age-related problems at present, I would say, is still a myth. Till evidence proves it, one should always treat it as caution or something which doesn't work. So this is from a GermH study, which is still unpublished data. But if you look at it, these are the eggs from 2 women. And one is the younger one, the left one, which I'm just clicking, and you could see the middle line was the aligned chromosomes. And as the meiosis resumes and the egg is dividing, so one is the polar body getting extruded, and the other one will form the pronucleus going forward when it fertilizes with the sperm. So all the chromosomes you could see were well aligned in the center, and that they proceeded across equally. But if we look at the other one, which is from an older woman, and you can see how the chromosomes are all scattered, you saw how it is all scattered. So some of the chromosomes may drag an extra chromosome along with it and result in trisomy, like Down syndrome risk. So chromosome segregation errors happens because of that. And this is all down to the single chromatids. What happens is in the nuclear DNA, the chromosomes, when they separate, and there are proteins, and that's what we have been looking at, cohesin and other, which bind them together when they separate. So they are all in the chromatids, in the single chromatid. So that problem cannot be overcome by the mitochondria, which is in the cytoplasm as such. So that's why the concept is less likely that it would work. And what we think, think there might be role of transcriptomics, that may be age-related, the protein that are produced rather than the mitochondria, because all our experiments, when we have looked at it, we have looked at lots of age of eggs, and we have been doing this technique and all our unpublished data so far, because we have to gather more evidence and more experiments to do before we publish it, has shown that it's the chromatids, not the mitochondrial DNA heteroplasmy, that is going to contribute to this. So that's why wait for evidence before jumping into offering all this kind of therapies, the augment and other types of it, treatment and in UK is not licensed as well for other indications. But I must admit there are other centers who have started offering this technique abroad, in Ukraine and in Greece and all, for recurrent implantation failure, miscarriages. But I want to make it clear, evidence is not there. So please don't jump into things which is not backed up by science. So all of us should have a common goal. How safe is safe enough? And what are the approaches to dealing with risk of ART medical treatment? Always comes along with risk. So this does not differ from medically assisted reproduction, and we cannot avoid this risk completely. However, we should make our effort to limit these risks as much as possible. That is, we should have responsible ART practice. And our common goal should be, we should be doing what is safe, what is ethical, what is legal, what is reproducible, not that like the therapeutic illusion, it works and next time when you try to repeat it, it's not possible. And for the right reasons, as I said, for mitochondrial DNA disease, it has been proven, but not for other reasons. So till it is proven the other way around, we shouldn't be using it. And this we owe to our patients and the future generation to come. So we should be working together for that. And I would like to end, I don't know how much time I took, but I get very passionate about this talk. But I would like to end by thanking all the team members. This is us celebrating our second Nature publication. You can see, it may not be very clear, but I've got orange juice, but the rest, all of them are celebrating with champagne. So our lab team members, this is Newcastle Fertility Center hosting in the Center for Life. The mitochondrial donation clinical ART team, as I've mentioned here, because as per the law, we have to have separate team who leads the mitochondrial donor program, separate one who sees the patients, the counselors, everyone should be there. The mitochondrial genetics team, because we cannot offer that treatment unless we have got mitochondrial DNA specialist and geneticist on board, and we have to do joint consultation with them, as well as the research nurses who helped us in this process for recruiting the donors as well, because that's again a requirement by the HFEA, and not the least the mitochondrial donors who come forward. Wellcome Trust Center for Mitochondrial Research is our center where I'm affiliated to as well. Newcastle University are the partnering bodies. I put my email address and Meena Fertility is my Twitter. So thank you, everyone. And I'm going to stop sharing my screen so that Rajan can take over and the rest of the team can take over. Thank you for listening.
Oh, excellent talk, Dr. Meenakshi. I hope I'm audible there.
Yes,
yes, you are indeed audible. Thank you.
Excellent talk, very comprehensive, very enlightening on the topic of mitochondrial DNA disorders and the various strategies we have available currently to circumvent those diseases, particularly mitochondrial replacement therapy currently to prevent the transmission of these diseases. I declare the session open for questions and answers by viewers. Do we have any questions, Dr. Amit?
So the one which I posted in chat box.
Oh, that's not relevant to the topic. I'll start off for Dr. Meenakshi with a couple of questions. One is, which of the three techniques you described for mitochondrial replacement therapy, whether it's a maternal spindle transfer or a pronuclear transfer or a polar body transfer, is being adopted more frequently currently at your center? And is there any particular reason for it?
Yeah. So, we employ, thank you for your question. We employ the mitochondrial replacement therapy technique that we employ is the PNT technique. Okay. The nuclear transfer technique. And that is because all of our proof of concept experiments and all was published in that one. Okay. So, so that's what we employ. We are doing research for MST, but we haven't published that data yet. And there are still few hurdles, as I said. It's a very dynamic process and the separation is an issue. So that will take some time, and polar body, still no studies have published human data yet. So that would come, but we have got research approval to look into polar body as well. So we are doing that. So maybe another 10, 20 years, we take long time to get ours, everything, do it ethically and scientifically correct.
Yeah, thank you very much. Another question that logically comes to my mind is that, as you have addressed it partially in your talk, the epigenetic influence that this entire thing might have on the original DNA makeup of the recipient nucleus by virtue of the procedure itself or by virtue of new mitochondria and the new original DNA of the nucleus, that is mitonuclear interaction. Do you have any data on this right now, any epigenetic, or do you conjecture would there be any adverse effects on the original DNA of the recipient mother?
Sorry, can you repeat the question again? I'm trying to understand the gist of the question.
The gist of the question is the epigenetic influence that might have adverse effects on the recipient nuclear DNA, epigenetic influence arising either by virtue of manipulative procedure themselves or by virtue of the introduction of the donor mitochondria into the cytoplasm, resulting into different kind of mitonuclear interactions, resulting into epigenetic damages or adverse influences on the original nucleus. I know that the technique is quite nascent right now and could not be a robust data on this, but what's your take on this? Will it have any adverse effect?
Thank you. Yeah, I understood. So, sorry that I apologize for asking you to repeat the question, but I understand the question. So basically, Rajan, what I would say is that the epigenetic modifications are, even in IVF embryos, and ICSI embryos, there have been studies looking into it, but a lot of clarity is still required. And this, as you rightly answered your question, that it's a very new technique. And that's why it's important that we look at long-term follow-up of the resultant babies, because from the research point of view, we have looked at some which looks promising and doesn't show any concern. But from the clinical perspective, we have to wait for that. So we have got a research approval, a clinical trial approval to monitor the first 25 babies for long-term. And then we would be able to see if there's any epigenetic modification related issues and conditions that develop in those. And we will be able to share that data in future.
Right. Thank you. Thank you very much. Do we have any more questions?
No, as I said, it will be a jaw-dropping presentation, so maybe half the public will be fast
I'll
asleep,
then sum up the
or
session
they
by
will be
saying
so
that
awe of the science that they don't know what to ask.
yeah, I'll sum up the session in a few lines that
Thank you.
mitochondrial DNA disorders, though individually are less common, collectively put a lot of burden on the society itself, and the prevalence is somewhere around 1 in 4 to 5,000, which is not less by any means at all. And there are no effective treatment modalities available once the baby is born or on the obstetric performance of the affected woman. And prevention has the best chance to prevent these inheritable mitochondrial diseases. And you elaborated very well in your talk of the various preventive strategies. I am sure this particular topic is going to be in focus in the horizon of the assisted reproduction technology for the coming decades to come. And not only would it be in the focus, it would be much more streamlined in the management approach. And we will have definitely a data emerging out of. And I congratulate you for being a pioneer in this wonderful field of mitochondrial DNA diseases and mitochondrial and their treatment or management by mitochondrial replacement therapies. Thank you very much for being our speaker today. And I wish you all the best in the work you are doing, and hope to hear a lot from you on this topic in future too.
Thank you so much for having me.
Yeah.
Thank you so much, sir. Thank you so much, ma'am. In fact, I will say that 40 to 45 years back, the type of technology IVF, which was in its nascent stages, or that was one we were looking forward to. Now, I think mitochondrial diseases, DNA diseases would be something which we'll be looking forward to. And congratulations to you for this pioneering work. And thank you for being a faculty for us.
Right. Thank you.
Thank you. My pleasure.
Yeah.
Thank you for having me.
Thank you. Yeah.
Nikhilji.
Okay then.
Thank you so much, sir. Thank you so much, ma'am. From AmCure, we are, really, it's a great pleasure to have both of you today. Of course, Dr. Vaidya as a wonderful host, and of course, you as the great speaker. Yes, we feel that it's an international icon today. So we started the icon event today, and thank you very much to each one of you. Thanks a lot.
I'll interrupt you for 1 minute. This is going to be a continuous series of webinars where we'll be having international icons of IVF. The first one, we had the honor of having Dr. Meenakshi Chaudhary as our international icon. And I promise we'll have wonderful icons of IVF who are actually shining stars on the international horizon of IVF. And all my thanks to AmCure Pharmaceuticals and the entire team working day and night to bring this concept of mine into reality. Thank you very much, Dr. Nikhil, Dr. Amit, and the entire team of AmCure.
Thank you so much, sir. Thank you very much. Thank you, ma'am.
Thanks.
Looking forward to see you again, in future events as well.
Right. Thank you, Dr. Meenakshi, once again.
Thank you so much. Thanks a lot for inviting me. Thanks a lot.
Most welcome. My pleasure.
Okay.
Okay, fine.
Bye.
Bye. Okay, Dr. Amit, leaving the meeting.
Right, sir. Yes. Thank you so much, sir. Thank you.
Recording stopped.
Are we still with the other team?